All right, ladies and gentlemen, welcome back. So we are going to wrap up our discussion about eukaryotic cells, just talking about some of the external structures that exist in these cells. So first off, let's talk about plant cells. So outside of their plasma membrane, they do have their cell wall. This is an extracellular structure. It is made mainly of cellulose, and then you've got other embedded polysaccharides, or other sugars, other proteins that are sitting in this structure as well. But the main component is that cellulose. And we find cell walls in your plants. But also remember they do exist in prokaryotes, they exist in fungi, they exist in some protists. The components to that cell wall might look a little different depending on what kind of organism, what type of cell you find it in. The definition here on the slide is mainly for plants, but things like prokaryotes, we talked about the peptidoglycan and the connection to the gram staining. So fungi, a lot of it's chitin. So depending on the major components, that may vary depending on the species that you're looking at, but overall, it is just this extracellular structure. It's one more layer of structure and support outside of their plasma membrane. And for a lot of them, it helps them to prevent excess uptake of water. So when we get to chapter 7, we'll talk about osmosis again, and you get into certain situations where water is rushing into your cells, and you need a way to prevent that from overtaking your cell's structure. If you can't do it, then your cell just explodes. And for a lot of these organisms, having this cell wall, one of its purposes is to provide them from extra protection from taking in too much water and just blowing up like a water balloon. They do have many layers, so if you take a look, you do have your primary cell wall here on the outside, a secondary cell wall here on the inside, a middle lamina, so there is some space connection in plant tissue, for example, between them. So they do have some multiple layers here. And when we talk about the division of plant cells and that creation of that cell wall, the vesicles that form, the cell plate that forms, there is some maturing of that cell plate into the ultimate structure of these multi-layered components to a cell wall once that cell finishes its division. So again, that primary cell wall is relatively thin and flexible. That middle lamina is that thinner layer between one primary cell wall of one plant cell to another, and that secondary wall is added between the plasma membrane and that primary cell wall. Something that does become challenging for plant cells when we're looking at plant tissue is that they still need to be able to coordinate function. And in order to do that, that means they need to be able to pass materials from one cell to the next. Their plasma membrane is still selectively permeable, just like we would see in any cell, but that cell wall structure is a little bit less permeable to material. So in order to have these connections, what they have evolved are these things called plasmodesmata, and they're basically little channels that punch through from the interior of one cell to the interior of the next. And so they perforate those cell walls to allow water, solutes, proteins, RNAs, that kind of thing, to pass from one cell to the next so that they can coordinate cellular functions so that the entire tissue works collectively for the plant as a whole, which is pretty cool. So let's go over to animal cells then. So plant cells, when they are in a multicellular organism and form those tissues, have those cell wall components to help hold them together. They've got the plasmodesmata as ways to communicate with each other. When we look at animal cells and multicellular animal tissue, we don't have that cell wall to hold us together. So what keeps us together? And that's where your extracellular matrix comes in, or your ECM. And we've got a whole bunch of things that go on here. Things like fibronectins, collagen fibers, proteoglycan complexes. A lot of these are carbohydrate protein complexes here that hold us together. And for our purposes, kind of think of it like Velcro. So you've got the base of that Velcro stuck to some sort of fabric on one side, and you've got all of those sort of loops, and then you've got the hooks on the other, and your other cell membrane would be on this side. And that's kind of what I like to think of it as. So it's going to help in the adhesion, keeping everything together. If you think about collagen fibers, if you know anything about them, these are kind of what gives your skin its suppleness, its flexibility. And if you look at older individuals, that collagen isn't as good. For whatever reason, it seems to break down in over our lifetimes, and we're not good at making more of it. Um, as we get older, and so that's where you get the wrinkles and the bending, and you're just not getting that flexibility as much. So that's part of that reason. Not only does it add for adhesion from one cell to the next in those tissues, but it's also going to help regulate some movement, as well as just communication components and just general support to the structure of a tissue. So just like you had those plasmodesmata in your plant cells, we also have junctions in between cells in our animal tissues. And you've got these tight junctions that tend to happen at the upper layers of tissue, and they basically form like a watertight seal and keep all of your tissue fluid inside your tissue, so you're literally not leaking fluid all over the place all the time. We have these desmosomes, which are sort of anchoring junctions, and they are kind of that idea of that Velcro and being able to hold everything together. And then we also have gap junctions, which are very similar to those plasmodesmata and will connect the cytoplasm of one cell to the cytoplasm of the next directly, as opposed to needing to go through your plasma membrane first. And so you may also come across these as communicating junctions or communication junctions, where we can send solutes and RNAs and that kind of thing between one cell to the next and just sort of expedite that process so that we can coordinate functions more quickly within cells in a tissue.